All right, thank you again to our presenters for their fantastic presentations. We'll now begin our question and answer panel to include all presenters and additional panelists joining as well, which I'll introduce in a moment. But in the meantime, we want to go ahead and invite attendees into the room. Please line up at the mics if you have questions. You must have some questions after those really informative sessions, right? Yeah, okay. So as I mentioned, we'll have all our presenters here, and also joining the panel, we have Dr. Yili Li, with Senior Pharmaceutical Quality Assessor with the Division of Product Quality Assessment 11, within the Office of Product Quality Assessment 2. And also Dr. Bryan Newman, a lead pharmacologist within the Division of Therapeutic Performance 1 in the Office of Research Standards within the Office of Generic Drugs. And also for those logged in online, if you haven't had a chance to submit your questions using the online or on-screen link and QR code, please do so now. And we'll answer as many questions as time allows. So let's go to our first in-room question here.
Thank you for the presentation. So for some of the peptide product that's approved way before the guidance is published, they may not necessarily have any unspecified impurities at 0.10% level as the specification. So would that play a factor in agencies' review when looking at the ANDA applications? Because if they can be allowed to be higher than that, then we are holding to a much tighter spec and the golden 0.10 number.
So yeah, you're talking about if you're talking about peptides. So for those peptides covered by the peptide guidance, yeah, usually we ask you kind of like, identify everything at or about 0.10% and identify control in the specifications. Also, there are some peptides not covered by the guidance, but however, if you look for PSG, the PSG will clearly state like this guidance applies to this product. We have similar expectation for the approval at the end for that type of product. Do you have any question for a particular product or just the general questions for the?
Actually, if you don't mind, I'm going to jump in here, having a little bit of work, not as much as Yili in the peptide arena. What we really look for peptides, while that's the guidance of 0.10, when it comes time to really doing the actual work and drug formulation, it's always the comparative analysis versus the RLD. So if you do your homework really well in P2, you're looking at the impurity profile of the RLD. And so if you see the RLD is having a higher limit than you would normally have according to the guidance, you can submit those 3 batches relative to the 3 batches that you have. And usually at expiration, you want to be doing that on the RLD, and you want to be doing that as far along as you can for yours as well. And you can provide a good justification, in which case the agency will consider that.
Great, thank you for those responses. We'll go ahead and move on to the next question in the room. Over on this side.
Hi, thank you for the presentations. I just want to note that the presentations that give challenges and potential solutions are extremely helpful to industry. And my question is related to some of the particle size data that goes into the applications. You know, we include all of these particle size testing data, and in module 2, where we do summaries, the agency has published some summary tables for doing particle size by MDRS, and I'm wondering if for particle size done by different methods, such as laser diffraction, Are you also looking to have us generate some type of summary tables for Module 2, or do you not care?
Okay, I answer in general. So in general, everything in the application has been shown and we should review everything.
Right.
So anything included in the P2 summary, yes, we look at it and review it. Any justification provided, anything we usually do. And if we have any concerns, we cite the deficiency to the applicant for more clarifications. That's in general. Did that answer your question?
I'm talking about the Module 2, like 271. I'm just wondering if you want summary tables for particle size along with what's in the Module 3.
Go ahead. Yeah, I think if it's helpful, definitely do that. I don't think it's a requirement, but if it's something that's going to help the justification, then you can do that.
And I may add here, I recommend everyone. So whenever you submit anything you may think about, okay, it will be shown or it's not important, provide a reviewer guide. Reviewer guide is really amazing that you can put all the documents with hyperlinks and includes everything you need to. So we usually use this as a very guide for us, help us a lot to find whatever we need. And summaries and tables, that helps also a lot. That's in general.
Okay, thank you.
Again, we'll go on to our next in-room question on this side.
Yes, thank you. My name is Michael Lichminat. I just wanted to say thank you, FDA and CBER, for having this training today. It's really informative and just gives the industry a better understanding of what the recommendations and expectations are. Just quickly, could be directly to Maria Flynn, Mrs. Maria Flynn, or just in broad for the FDA panel. I just wanted to ask, is there any agency updates to the IIG database, specifically to improve excipient toxicity level in pediatric patient within the oral suspension type, maybe oral suspension specifically dosage formulation, or in general?
I love the question. Thank you so much. So something I didn't have time to go over a lot of times with taste masking and a lot of the different formulations, like for suspensions, there's a lot of intricacies that can occur, and in each excipient can have different impurity profiles. And so he was talking about the IgG, which is the inactive ingredient database. And so there's a database, and you have to look at it not only for the actual exposure, but you have to think about it as the combined exposure of your different excipients. And specifically, in a lot of the excipients that are used, there's going to be trace amount of impurities, and we have a good sense of what those are. And it's really important to know your excipient impurity profile, because some of them are of a cohort of concern. They do have toxicological. So to your end, the database is as current as possible. So you do that, but when you do it from a holistic perspective, because there's going to be multiple excipients. And sometimes when you look at them, like for instance, flavoring, it's very important to get that information from whatever vendor that you're doing, like Tutti Frutti or something. For instance, there's a case today I'm thinking of in my mind where there was an issue with benzaldehyde, which is a cohort of concern. And it was in multiple excipients, not excipients, but ingredients of the flavor. And had they not looked at that profile, they would not have known that it exceeded what was normal and healthy and safe for use. So to your end, the database is as good as we have right now. And I believe they updated it. If you obviously, if there's something that's more helpful, and if you need to, please reach out to the agency so that we can include the information that you have or information that you're concerned with. And specifically, if you have something that you're trying to do and you're concerned about it, go ahead and submit those control correspondences to us. And if necessary, not in oral liquid situations usually, but in the more complicated situations, you want to use our pre-ANDA. Make sure you go through that pre-ANDA process if there's something really interesting and fun going on. And the sooner you communicate with us, the sooner we can help you and be proactive.
For sure, thank you.
Fantastic. We have another question here on this side of the room.
Okay, so the impurities above 0.1% should be ID'd for peptide, right? But to what extent the mass spec should like the identification should be. Do you expect like full structure and sequence information, or can we just do a mass spec comparison and match to the RLD impurities?
Oh, yes. So that's a good question. We go back about the impurities. The general idea is, we encourage you identify everything at or about 0%, and then, do extensive comparison with the RLD. In that way, like, we understand what's the differences between your ANDA and the RLD. If you identify I think for certain impurities, only retention time, you say, the same as RLD impurity, that's not sufficient. So we usually ask you to provide further characterization. For example, you say the same molecular weight or like MS fragmentation, the same as RLD. Then if you observe the RLD level is higher than your end product, then that means your limiter is qualified. However, if you observe the level in your product is higher than the RLD, then you may probably think about like immunogenicity mitigation.
Thank you.
All right, if we don't have questions in the room at the moment, we'll move to an online question, but I still want to encourage everyone here, don't be shy, still feel free to come up to a mic if a question pops in your head. So we'll go to our first online question.
All right, this is for Dr. Nashwa El-Gendy. How do you deal with polymorphisms in DPIs? Yes, that's correct.
So for DPIs, this is a powder. So powder characteristic is very critical here. For the API and the carrier polymorphism for the API is very critical, and applicant should show if there is any polymorphism, they should have a testing for this polymorphism to be sure that the consistency polymorph used all over the drug substance lots. They have to provide all the chemical characterization or evaluation, PXRD, TGA or DSC, all the data for at least 3 lots of the drug substances with the polymorph that's consistent. Also, it's recommended to include polymorphic phase testing in the drug substance specification. For the drug product as well, the batches, it should be tested for any change in polymorphism throughout the shelf life of the drug product. This information we are looking for all the time.
Thank you for that response. Again, if anyone else has questions in the room, please come up. But in the meantime, we'll go ahead and go to our next online question.
All right, this question is directed towards Dr. Bryan Newman. Recently, the FDA has published PSGs for DPIs that included other alternative in vitro studies that could be used for bioequivalence or product characterization. Can any of these studies be used to test the RLD or a proposed generic?
So that's a good question. I think, so what we can say now is that, of course, what's currently posted in our PSGs are current recommendations, but as was discussed yesterday during the presentations on alternative approaches, we're obviously been looking at both what study designs for alternative approaches will work for both MDIs and DPIs. So I would say that certainly some of these tests can apply for DPIs, and there was a good slide to show that across even with the MDIs, a lot of this depends on the characteristics of the product. So if you're considering this particular DPI, the API itself, and its own characteristics, you can try to discern what types of alternative approaches might be applicable for that. So with that in mind, of course, you could then develop a alternative approach and provide that in a pre-ANDA meeting request to get feedback from the agency. And certainly with considering this, I'd also point out that the PSG forecast list provides a good indication of how we plan on working through and considering the different PSGs, which ones needs to be revised for the alternative approaches. So if you're considering developing a product and you have this question in mind, I'd encourage you to take a look at the forecast list to see when we might be providing guidance and consider how you want to develop your program at that point.
Thank you. So it still looks like we're a bit shy in the room, so we'll still move on to our online question. Yep.
This question is directed towards the panel in general. If an API cost per milligram is very expensive for an ANDA applicant, does the FDA accept ANDA applications with smaller batch sizes other than the recommended ones? What is FDA's expectation?
So I'm talking from the, for example, for the complex drug product, we usually recommend 3 batches on the commercial scale. However, also again, we may accept 1 batch at a large scale and 2 batches, 1/3 of the commercial batch scale. And again, if there is any questions like this, we highly recommend the applicant to use all the FDA communication channels during the development stage through the pre-ANDAs or CC with all the questions, and we are happy usually to help them with any with clarifying any question they have. Thank you.
Thank you. So we'll go on to another online question.
All right, this question is for Tina Jiao. What are the recommendations for the samples used in comparative studies?
Yeah, for the test product, we ask for at least 3 batch of the test product manufactured from at least 2 lot of API tested on or near the at the release and at the end of the shelf life. And also we ask for at least 3 batch of the ROD, I mean, if available, at a different age before the expiry. And also in your submission, we recommend you provide a table to list the test product, the batch number, the API lot number, the manufacturing date, the expiration date, and testing date to help us to figure out the age of the sample. Yeah, that's it. Thank you.
Thank you for that response. Do we have another online question?
Yes, so this could be a follow-up for Tina Jiao and as well as to Dr. Yili Li. If I performed the comparability study aged and near release, it is recommended to update the study with samples aged at the end of the proposed shelf life. But which test needs to be updated? Only impurities profile comparison? Or do we need to also update the aggregate test? Or which tests need to be updated?
Yeah, you know how to answer. Yeah, I think all of them should be updated. Yeah, thank you.
Yeah, I just want to add, the characterization not only for impurities, but also for like high order structure and aggregations. So the study should be tested and should be performed near release and at the end of shelf life. We want to say like at the end of shelf life, your product behave like properly.
Thank you again for those responses. And again, reminder to those in the room, this is your chance to come up and ask a question. All right, we've got someone on this side of the room.
I have a question for the panel. So you talk about accelerated testing. And in practical, there's a lot of different rules used, maybe Arrhenius equation or the 10-degree double rate, those things. But we know that's not universal for everything. What thought accelerated testing you have to unify those things?
Can you repeat the question? Just I missed the
accelerated testing.
Oh, accelerated.
Yeah, accelerated testing for the shelf life. Like it's 2 years shelf life, you do 6 months at higher temperature. And what temperature you're going to choose, what time choose you based on some acceleration equations. Today we use some simple rule like Arrhenius equation or 10 degree for double rate, something like that. My question is not everything follow that equation exactly. And what's general thought to case by case to do that?
I can go on for this question. So I recommend you look at ICHQ1E. It states very clearly what situation, for example, you have, if you fail accelerate, what you're supposed to do. Usually you need to start in intermediate stability. And also if intermediate stability increases significantly, what you're supposed to do. So there's a very clear decision tree in ICHQ1E. If you follow that, it will be clearly stated what you're supposed to do. And if the long-term stability is room temperature, what you're supposed to do. However, if it's long-term stability is refrigerated, what you're supposed to do. So I recommend you just go follow the decision tree in ICHQ1E. Did I answer your question?
Yeah, I agree with Yili. I feel like the guidance is very clear, but I want to bring up one special situation, and this doesn't happen. I have a lot of work done in the PEPFAR space, and sometimes in the PEPFAR space, we have zone 3 and 4. So when dealing with things that we know are going to different climates, such as very humid and, in the Southeast Asia or in Caribbean or in Africa. In those situations, those are a few deviations that we would say away from the guidance. But in those situations, then they would have a higher, like 30 degrees and really high humidity and sometimes. So those are the few exceptions, but for the most part, they do follow the guidance in terms of if there is an issue with the accelerated, then you would follow the guidance.
Thank you. And now we have a question on this side of the room.
Hi, it's me again. You recommended the orthogonal method for the characterization, right, of peptides. So say if we have two methods, we try one of them is not working, like feasibility, it's not feasible. So should we just keep trying till we have a second method, or would that like one method be acceptable in that situation?
So yeah, usually we recommend like orthogonal method. So it's kind of like in cross-checking whether your results are like comparable to each other. Most of the time, so I don't see. Yeah, I think if two methods are not working, you should try more different type of method. And if you really exhausted all the technique and you only found one method working, I encourage you provide like a clear justification for everything why, like state clearly why this method is not working, what's the problem with that. I would say it will be case by case, but in general, yeah, we do recommend that you have an orthogonal method.
So like say if like for aggregation test, right? Say
Right.
if one method find no aggregation and then we do a paper-based like a risk justification, the other method, for example, SAC mouse is not working, do you think that can be approach we can take? Very specific, but
Yeah, it's really like I cannot give you straight answer, like because it depends on the product, also depends on your result. As I said, if you think no aggregation detected with one method, you definitely need to provide clear information, like whether your result is. Yeah, whether like your result is correct, so you definitely need to check. And I don't see, yeah, so for peptide, usually we recommend you do the like high order structure also. Actually, if you have aggregates, sometimes NMR is can also be a good indicator, like whether there's aggregate. So NMR, high resolution, like two-dimensional NMR can like double check both the highest order structure and the aggregates, whether there's an indication. So I don't say like, I mean, so far I don't say like none of the method working for certain thing.
thank you.
All right, we have one more question here.
Yes, I'd like to know, will the agency grant 36-month dating on an initial application, an ANDA, for a complex product if you have the 36-month data for both your product as well as the brand? And just to be clear, the size of the manufacturing that you've submitted is the size that commercial will be at. It's the commercial scale.
So your question is if the applicant provided 30-month stability data for his product and also the brand in the commercial scale, in the commercial manufacturing site.
Yes.
What's the question in this case?
The question is, would you grant 36-month expiration dating?
It depends on the data. If the applicant propose 30 months shelf life, we will work on that to see if it's acceptable or not based on the data provided. If the applicant provided 24 months and they have 36 months, but they propose 24 months, we'll not tell them, yeah, you have until 36, you can extend it now. No, it's the applicant decision. But the data provided based on what the applicant provided, what he is proposing. You having your application in P8, you're proposing 24 months shelf life. This is our shelf life. Extra data, we review it for sure, and if it's adequate, but we accept 24 months based on the proposal provided to us.
Right. Okay. So you can, if you have 36-month commercial scale, it seems the agency would take that into consideration. And
It's reviewed. Everything is reviewed, but at the end, the product approved based on the proposed by the applicant. Applicant proposed 24 months. That's the shelf life. Yes, if it's everything is fine, it's fine. If applicant would like post-approval, they can provide a request to extend the shelf life or whatever based on the data already provided. Or if they feel that the product already up to 36 months adequate data within the specifications, they can request to extend the shelf life to 36 months.
Okay,
It's up to the applicant. Welcome.
All right, thank you so much. Looks like that's all the time we have questions for, or all the questions we have time for. Thank you again to our presenters and panelists for all the excellent information and insights. We'll now be taking a break which will resume at 11 a.m. Eastern time.